happen now. Okay, let's wait a few more minutes. That more people come. Because we had quite a few registered participants, and let's wait a minute or two, and then we can start. Yeah. Okay, I think the numbers are converging. So let us start. Yeah, so good morning or good afternoon, sorry, at least in our part of the world, and good evening in the part of the world of our speaker. Welcome to our NITheP webinar. And because our speaker is in Australia, the time is not the usual time, but we had to find a compromise that is acceptable to both parts. Yeah. So today's speaker is Dr. Kavan Modi, that some of you might have met because he visited us in Durban. He's been in Durban before, but now he's based at Monash University in Melbourne, and he's locked down at the moment, like the rest of us. Yeah. So Kavan, thank you very much for accepting spontaneously to offer us a webinar. Because after we saw your recent preprint, I thought it would be a great idea to ask you, and you kindly agreed to share your new results with us. And I know that you mentioned that it's not a quantum biology talk, but it's a talk about effects that that have interested us in the past and at the moment, and we are really curious to hear what you're going to share with us now. Thank you very much, Kavan. Sorry, let me stop sharing the screen. Voila, and over to you.
Okay, thank you, Francesco. Thanks for a kind introduction. I'm very happy to be here. Let me see. This might be it. Let's share. Yes, okay. So you can see my slides?
Yes.
Perfect. Wonderful.
Thank you.
All right. Thank you, everyone, for joining me, for joining this seminar. So I'm going to talk about stochastic processes, complexity theory, and a bit of neuroscience. And this project started maybe 3 or 4 years ago. Or maybe not this project, but this adventure started 3 or 4 years ago when one of my colleagues at Monash University, who's a neuroscientist, Now Suchia, who's also an author on this paper, he and I happened to be attending the same event in the mathematics department, and we got talking, and we realized quickly that we had quite a lot of overlap. And we started thinking about problems. And along the way, we came up with this problem. That was the first attempt at doing something together, and it worked out quite nicely. And there's a broader effort in Melbourne, between Melbourne University and Monash University, to do some serious consciousness research. And this is the logo on the top right-hand side of the screen. Melbourne Monash Consciousness Research. It's a group of physicists, psychiatrists, philosophers, neuroscientists. Some people do dream studies, etc. So it's quite broad. And T-Lab is the lab run by colleague Now Suchia, who's a neuroscientist. And my group is called Monkeys, Monash Quantum Information Science Group, and given the link below. So mostly what we do is quantum information science. Okay, so let me go to the next slide. I just give you a quick 2-second introduction to what Monash university does in terms of physics. So it's a lot of cold atoms and condensed matter physics, imaging physics, and then there is astrophysics, including LIGO and people at CERN doing particle physics, and then lots of imaging physics. And I bring all this up just in case if somebody's interested in joining Monash University for a PhD or something. There are lots of options, and I just wanted to give that option, give that overview. And of course, there's quantum information, which is just me and my little group. Hopefully we grow in the future. This is the group. This is quite an old picture of the group. I've only listed a few people here. So Roberto Munoz is a PhD student who's led this project along with Aidan Zetwick. And Felix Pollack, who's on the left, he is the postdoc in the group, and he's also played a key role in developing these tools. So mostly what we work on in our group is complex quantum dynamics. We also do a bit of quantum control and quantum sensing, quantum thermodynamics, and a few little things here and there. And today I'll talk about one of those little things, which is complexity and neuroscience. So this is the first time I'm giving a talk on this topic, and this is also the first time I'm giving a Zoom talk. So I hope those two first go okay. So do let me know if you have any questions along the way.
You're doing just great.
Oh, thank you, thank you. It's hard to do these talks without any audience participation, at least visually. Anyway, so recently we published this paper in Physical Review Research, and it's got quite a long title and quite a long list of authors as well. And so some of the authors, Dr Kavan and Bruno van Swinden, are the two experimentalists who did these experiments. Naosu Chia and Angus Leung are neuroscientists, and then the rest of the crew, Roberto, Aidan, and Felix, are from my group. We did the theory and numerical analysis here. So let me tell you about the experiment. The experiment took place a couple of years back already, and so we just got the data and we played with it. And so I'll give you a brief description of what the experiment was. And you know, this is a theorist version of the experiment, so keep that in mind. So let's say we have a brain of a fruit fly. So this is a transverse view of it. Those big bulging things, unable to point at things. Is that right? I don't see a mouse cursor. Anyway, so those big bulging things on the top and the bottom are the eyes, and then rest of it is the brain. And in the previous slide, you can see that a little bit better. There's more structure there. Neuroscientists could tell you a lot more about it. For me, it's just a big blob. And then the experimentalist, this is Dr Kavan, who's one of the authors on this paper, also the first author of the paper is listed on the left. He inserts a probe into the eye, and that goes halfway through the brain. And this doesn't kill the fruit fly, surprisingly. The probe is very small. It's, I think it's 2 millimeters long. I mean, you know how small fruit flies are, so 2 millimeters even seems too big. So this probe goes in. It's a very small probe. At the same time, it still has 15 channels. So these pink dots on this needle are different channels. And what these channels do, so on the right-hand side, you see this big black and red blob. That's the eye. And the probe goes into the eye. And what the channels do is they detect local field potential. So in other words, they detect the potential at the potential electric potential as it changes in time. 2 sets of experiments are done where first the fly is awake, and then fly is given some anesthesia, which is isoflurane, and then there's a way to check whether the fly has fallen asleep or has been anesthetized by hitting it with some air puffs, and one can visually check whether the fly stops responding. So once the local field potential is measured, which is measured, I believe, for 18 seconds at 25 kilohertz, I believe, which is then downsampled to 1 kilohertz. So we get about 18,000 data points, binarized data points. And the binarization is taken by doing time binning, and also by making ones and zero around a median. So the field potential is binarized, and then we get to do whatever we want with it. So we get to perform some sort of an analysis on this binarized data. And I'll tell you about 2 of these processes. In fact, there's a third one as well, but I won't talk too much about it. So the paper written by Dror and Bruno, the experimentalists, what they actually did was they looked at, so they did a bunch of experiments with 13 flies, each for 18 seconds, where they measured the local field potential in awake and then in anesthetized for each fly. So that's total of 13 times 2 sets of experiments, if you will. And then a power spectra analysis is done. Now, this is something we're very familiar with in physics. What you do is you look at the signal and you look at the time difference between the signals. So let f of t be the voltage at time t, and let f of t minus tau be the voltage at time t minus tau. And then we can integrate that over time t, and then take the Fourier transform of that. That gives us the power spectra. And on the left-hand side for awake, and on the middle for the anesthetized fly, power spectra at the center of the brain and the periphery of the brain are compared. It's kind of hard to see that there's a difference, but if the 2 signals are subtracted from each other, you can clearly see that at high frequencies, there is a difference between awake and anesthetized. So this power spectra analysis is already able to identify whether the fly is awake or anesthetized. Now the question is, why might you care about that? And there are multiple reasons for it. A practical problem in neuroscience is to differentiate between a patient who is in a locked-in syndrome or in coma. So a patient who's non-responsive could very well be aware of everything that is happening around them, but may not be able to signal the outside world that they are aware. Or they might not be aware at all. And clearly there are ethical issues here that you have to account for. And you know, we would like to know how we could help such a patient if they are indeed awake and aware of what's happening around them. Another issue is that we'd like to be able to differentiate between different conscious states or different conscious arousal state, where we are, where a subject is in fully awake state, or partially awake state, or completely anesthetized or a sleep state. So these different conscious states would be interesting to identify in order to have a better understanding of neurophenomenology. And so these are the reasons for looking at these things, and just to understand the brain as a physical process, or the neural signaling as a physical process as well. So a popular way of doing this at the moment is something called integrated information theory. And integrated information theory is built, axiomatically built to deal with consciousness. I'm not an expert on this, but my colleague now, Suchiya, who runs TLab, he is an expert at this. And I've taken these images out of, I think something Giulio Tononi, who's one of the pioneering researchers pushing for integrated information theory. So these are the sort of the important elements of consciousness. And then these things are associated with cause and effect. And this is where a subjective science of consciousness meets something objective, like physical processing or information processing. And this is where Now and myself started discussing with each other. Now, I'm not an expert on integrated information theory, but Now's group has applied various tricks and techniques from integrated information theory to this exact fly problem. And they're also able to differentiate between awake and anesthetized and other phases of fly. By the way, this might seem like an easy thing to do, and in some sense it is, but what we would really like to get to is be able to differentiate between two different conscious states that have the same level of awakeness associated with them. And maybe I can talk about that later. So while we're discussing integrated information theory, my group has also been looking at stochastic processes. And so we thought to apply some new tools that we've been learning about from stochastic processes to this problem. So let me give you, I'm preaching to the choir here, of course, but let me just say a few words about stochastic processes and the way I see them. And this is all classical. So of course, a nice example of a stochastic process is traffic. You know, where if you have a grid of intersections, then something happens if something happens in one intersection, then it affects the whole grid. Another nice example is, say, the stock market, where let's say price of a stock is at the beginning of the week or at the end of the week is something, and we want to predict what happens in the future. And this is a good way to describe a stochastic process, because we can write. It's sort of easy to say it's easy to define a stochastic process in this way. So let's imagine that we wanna know the price of the stock starting tomorrow on Friday, all the way up to. Starting on Monday next week and all the way to Friday. So the time is reading from right to left. So Friday being the latest event. And there are two possibilities. Say that on Monday the stock will be $30, then 24, then 30 again, then 30, and then 11. Or it could be 20, 14, 20, 14, 20, 14, and oscillates back and forth. So formally, a stochastic process, I think that went the wrong way, is the joint probability distribution of all possible trajectories that stock can take. So all the possible values that the stock can have during next week forms a probability distribution. So there's probably a very low probability that the stock will be worth $1 million or $1 billion. So it vanishes quickly. So there is the distribution is peaked around some reasonable values, and that time order distribution is the stochastic process. Now, this stochastic process has to have some nice properties. So for instance, if we don't observe the value of the stock, say on Wednesday, that shouldn't matter what the distribution is for the rest of the week. Not observing the process for a day doesn't change the process. And this is known as a Kolmogorov consistency condition. And in principle, we can think of the stochastic process as a continuous probability distribution. So imagine that P sub t on the left-hand side in the bottom box is the value of the stock continuously, and then it also contains the value of the stock for 5 days on average. So in other words, I take the daily average, or over 4 days, or over 2 days, and so on. Now, all this formalism is fine, but let's say, what can we do with it? Well, the whole point of a stochastic process is that we'd like to predict the future. Right? I mean, we might not be able to do it deterministically, but we'd like to maybe do it probabilistically. So a process is called non-Markovian when we have to, in order to predict the future, in order to predict the price of the stock, say, next Friday, if we need to know what its price was on Monday, Tuesday, Wednesday, and Thursday as well. So if the stock had taken the red trajectory versus the blue trajectory, then if it means something different going forward, then the process is called non-Markovian. In other words, we have to account for some past, some amount of the past before we can predict the future accurately. In contrast, the process is called Markovian when we only need to know the current value of the stock, and then we can predict the future. So we can get master equations, by the way, from things that are Markovian or that can have slight level of non-Markovianity. So if you look at it in terms of stochastic processes, non-Markovian processes where we allow arbitrary conditioning are the most general set of distributions, the family of distributions that contain endpoint correlations. Master equations are the ones that contain 3-point correlation functions, and Markovian processes only have 2-point correlation functions. And then there are also random processes which only care about 1-point correlation function. In other words, the future doesn't depend on the current state as well. Okay, so with those formalities, I want to talk about modeling stochastic processes. And what I mean by that is I want to construct, so if I've got a non-Markovian process, can I construct a Markovian model for it? These are so-called the hidden Markov models. So let's consider a coin as a process. And coin, usually we think of it as a Markov process or a random process, but let's assume that the coin is correlated. And what we'd like to know is that, having seen the coin heads and tails a few times in the last few tosses, what's gonna be the future? And it could be that we have to know the coin has memory that goes back to 4 or 5 steps or 3 steps. We don't know. So how do we deal with that? Well, we can call the memory as just keeping track of what the coin has done in the past, and we can conditionally check whether the future depends on those past outcomes by tossing the coin a whole lot of times. And then we'd like to talk about how complex a process is. Now, suppose I have to keep track of the coin. I don't have to keep track of the coin. It's completely random. So this is the first depiction on the left-hand side. This is a Markov chain where coin has a single state and it just goes into itself. And so it's a random process. So I don't care whether the coin is, what the current state of the coin is. Makes no difference. I toss it. Sometimes I get heads. Let's call the pink arrow is the outcome heads. And the green arrow is the outcome tails. And I don't have to worry about what happens. The next occurrence will be completely random. Or I could have a situation where the coin goes between heads and tails in a deterministic fashion. And there, in that case, I have to know what the current configuration of the coin is in order for me to determine what the next configuration is. Or it could be possible that the coin is gonna be heads or tails in a more complicated way. And I have to remember the history for a finite number of steps. And so keep in mind that the complex process on the right-hand side is still a coin, no matter which state I'm in, whether I'm state F or I'm in state D. The only outcome is either a green arrow or a pink arrow. In other words, I only get heads or tails. But depending on what I get to another state, and then the statistics of what happens next are different. And I can also have things that go back into themselves. So this is called a hidden Markov model. More precisely, it's a unifiller hidden Markov model. So I'd just like to remind that coin here is doing nothing more than heads and tails, or zeros and ones, just like our fly data. It's just a binarized data, but it can have a highly complex structure depending on how much history there is. So if the longer we have to look back in order to predict the future, more complex the process is. So how do we do this? Well, so what we have to do is we have to look at a set of past sequences. So we can partition all the sequences that occur in the past, and we can call them past A, past B, past C, and so on. And then we can divide them up. And we can divide them up in the sense that suppose past C and past D lead to the same future, then we can put them together. And if past E and past F lead to the same future, then we can put them together. This is called Occam's pool. So it's a way of simplifying. The dependence on history. So we can form equivalency classes when future does not differentiate between certain set of past. So let me be a bit more precise with that. So what we can do is then we can label these past as, we call them causal states. And these causal states imply distinct futures. Probabilistically, but nevertheless distinct futures. So let's be a bit more precise. So let's say that I only need to worry about memory of last step. So I just need to know what my coin's configuration is. So then I only have to worry about two past, whether it's 0 or 1, what the current state is. But perhaps I have to worry about not just the current state, but also the last state of the coin. Then I have 4 possible paths. And this grows exponentially in the history. And this is why complexity of a non-Markovian process grows rapidly as you have to go further and further back. So for 3 steps, I have to worry about 8 possible paths. But it could also be that not all of these 8 possible paths lead to the same, lead to distinct futures, right? So it could be that observing 101, 110, and 111 always give me the same future. So I could have a causal pool that looks like this, where some of these paths are put together into a single little pool. And by doing this, so if I let's say I want to construct a model for my process, and I want to go back to 3 time steps and look at the history, then I can, for that much memory, accounting for that much memory, I can construct a hidden Markov model that is minimal in its complexity by grouping these paths together. And there is a way of doing that. There's an algorithm called causal state splitting reconstruction, or CSSR, that was developed by Shalizji and Crutchfield in 2001. And that algorithm you can download, and you can stick in time series data. You can specify how much memory you want to have in your model, and it gives you something called an epsilon machine. And what an epsilon machine is just a hidden Markov model that is minimal and is unifiller. So let's say I have some binarized data of coin tosses, and I say I want to go back and look at the history of, say, 3 or 4, and then I give it the time series data. In other words, a sequence of coin tosses, and tell it a number of memory number for memory length, and then it gives me a hidden Markov model, which is called an epsilon machine. And on the right, I have posted a picture of Occam, and the philosophy here is of Occam's razor, that a simplest model is then able to describe the process the best, or somehow that is an appealing model for the description of the process. And then you can do some nice things with this epsilon machine. For instance, you can compute a complexity for it. So these letters A, B, C, D's, E's, and E, F, those are called causal states, and those are you know, the states that the process can be in. So at a given time, you might be in state B, and from state B, you're allowed to transition to either E or F. And then once you're in F, you can go back to B, or you can go to D, and so on. And this is a digraph that is connected, and you can keep track of how you travel on this graph. Now, what that's actually done is turned a non-Markovian process into a Markovian one. That's what it means for it to be a hidden Markov model. It is a Markov model for the non-Markovian process. And so because it's a Markov chain, you're always gonna be oscillating between these 8 states. Now, of course, you're not gonna be traveling between these states equally. So you'll spend more time in some states over the other, and that allows you to compute a notion of complexity. So we can ask, we can define a statistical complexity as the probability for spending, the probability of being in ith state in the stationary sense, because it's just a Markov chain, so you can compute a stationary distribution. So the probability of being in ith state, log of that times itself, and you just compute the Shannon entropy for that, and that tells you the complexity of that process. Now, there's something nice about it, because this allows you to compare, take 2 given processes. So let's say you take the stock market and you take the weather, and you can ask which one is more complex, and you can compare the 2. And what does it mean? Well, it means that given either of these stochastic process, which one is going to be more demanding for purposes of modeling on a computer? That's all that says. And higher the entropy means that the more states you have to account for. And lower the entropy, the fewer states you have to account for. So the communication cost is well understood in terms of information theory. There are other entropies that we can associate. For instance, there's a topological entropy, topological complexity, which you can associate with the complexity of the graph. And we'll see a little bit of that in a second. Just to give you some intuition, a random process is going to have zero complexity. And a Markov process requires knowing the current state. And then you can ask the question how often the current state occurs, and then you can take the probability. So if the current state is almost always likely to be, say, 0 or 1, and very, very rarely you're going to get states 2, 3, and 4, then those almost don't matter. It's like a binary process. And then in general, you need to know the causal state. And that's because taking a non-Markovian process, you've Markovianized it by constructing a hidden Markov model. And all you need to know is the current causal state, and that is enough for you to predict the future. I'm happy to answer questions along the way if there are any. Just feel free to disturb me. I know I'm introducing quite a few concepts that maybe not everyone is aware of, so happy to be interrupted.
Thank you.
Okay, so let's apply these ideas of complexity and stochastic modeling to the fly. So as I said before, from the fly brain, we're getting this voltage, and that voltage is binarized. And now I'm going to construct a model, a stochastic model for this output of zeros and ones. And I want to consider length of 3. So I'm going to highlight code words of length 3. So I start with 0, 1, 1. So I start with on the state 0, 1, 1, and then I slide 1 to the right, which gives me to 1, 1, 0. So I see that there is I draw an arrow going there. And then I slide 1 more, and I get 101. So I draw another arrow. So those connections are possible. And then I keep going. And then as I keep going, I keep drawing arrows that are associated with all the allowed transitions. And if I do this long enough, I'll see all possible transitions. Not only that, I'll also see the probabilities for all of these transitions, because I can ask how often do they occur, right? So I've done the sliding a million times. How often did I go from the group that includes 0, 1, 1, 1, 1, 1, 1, 0, 1 to the group 1, 1, 0, and 0, 1, 0. It's a little bit awkward to talk about them like that, so I may as well call them something else. I can group them, and I can call them A, B, and C. C1 and C2 look distinct, but then if you look at the probabilities a little bit closely, you can group them together further. And then here's a little bit nicer picture of that same process. So given that I'm in state A in a stochastic process, the next outcome, which is going to be 1 or 0, and I'm going to slide my little highlighted region, that will take A into B or into itself. And then B can go back to A, or it can go to C, and then C can go into its own self or go to D, and then D can go to B, or it can go to A, and so on. So those are the rules. Now, notice that not every, so this is a memory length 3 state, and there are, the total possible states you could have is 8 but not all of them occur distinctly. So some of the histories are carrying the same information. And this, in fact, is a hidden Markov model for one of the flies. So if we take fly 1 at the central channel, so the center of the brain at memory length 3, we in fact do get exactly that hidden Markov model. Well, actually, we get 2. We get the model on the left for the same fly, same memory length, and we also get the model on the right. What's the difference? Well, the left fly is anesthetized and the right fly is awake. And so you clearly see that the topological complexity of these two are very different. The one on the right seems to have a lot more going for itself than the one on the left. Well, it's the same fly, same region of the brain. Nevertheless, the brain is doing something quite different here. And the statistical complexity is also different. It's higher on the right. So this was really the first thing we observed. And so then we tried to study this systematically. And what we did was, we looked at all the flies. This is fly average. There are 13 flies. We looked at all the channels and all of and different memory lengths for awake and anesthetized. And we computed the statistical complexity. And so red is higher and blue is lower. So we see more complexity at higher memory length. Lambda, which is on left-hand side, is the memory length. So we're accounting for memory length of up to 11. We can't actually do any more because we're data limited. We have only 18,000 data points. We would need a lot more data points if we wanted to go higher. We probably couldn't go higher more than 15 or maybe 20 at most because of computational resources. And we're looking at different channels. So different channels correspond to different places in the brain, 1 being the center of the brain and 15 being periphery of the brain. And we clearly see a difference between anesthetized and awake. And this is averaged over 13 flies. Now we can take this 2-dimensional plot and we can turn that into a 1-dimensional plot by either, so we take the differences in statistical complexity between awake and anesthetized, and we can plot that as a function of memory length, or we can plot that as a function of channels, so in other words, across the brain. And we see a little bit of oscillation as we scan across the brain on the bottom plot, and we don't quite know why. And in fact, if you look at here, you see that there's some periodic structure here as we go from left to right in both of these plots. We don't really know why that is. And we have no idea how to even explain that, but we've seen it over and over and over. And it's quite strange because there's a lot of processing already happening and we don't know what could retain this periodicity. But the interesting plot is the one on the top where the statistical complexity, the difference in statistical complexity between awake and anesthetized seems to be growing as we consider a more memory length, and it's significantly higher for awake, statistically significantly higher. Now, of course, this is statistics and biology, so if you look at the gray lines on the back, which is for individual flies, not all the flies we see the same pattern, but on average we do. And we do some careful statistics that our colleagues in neuroscience know how to do in order to make sure that we're not fooling ourselves. So we do hypothesis testing and so on. And I'll get into that in a second. But before that, I want to talk about asymmetry in flies. So and this is probably the most exciting result in our paper. So let's take a time series of data, right? So we have some binarized time series, zeros and ones. And what we can do is we can construct a hidden Markov model by considering that time series. And then what we do is we turn that time series into an alphabet of causal states. So knowing the causal states, we know, so for instance, if we're in state A, by the way, I wrote that randomly, so they may actually not correspond to the graph below. But say if we're in state A, and if we go to state D from A, then we know that we emitted a 1 because there is a green line connecting the two. So we can construct the causal state and causal alphabet, causal state alphabet, and that has all the information about the hidden Markov model. All the statistics are there. But there is nothing preventing us from reading time series backwards and constructing a completely different causal state, which we can call a completely different causal model for it, which we can call M minus. And in fact, we can take the causal state alphabet of the S minus and S plus, and we can combine them and run CSSR and construct a joint machine that contains both of those information. Now, you might think that why would you do all that? And in some ways, this is all for fun, but there are one can attribute some nice meanings to it. So Jim Crutchfield wrote several papers on this, and you can define a notion of irreversibility in the stochastic process by considering the difference in the statistical complexity between the two. And what that says is that that given the time series, it might be easier to model the stochastic process one way in time over the other. There's no reason to assume that it's going to be the same. So there is a notion of irreversibility already there. And I'll discuss in a few minutes why might we care about this in the current context. We can also describe something called crypticity. And crypticity is an idea that it quantifies the amount of information that one would need in order to synchronize these two machines. In other words, suppose you give me S plus or M plus, how much extra information would I need in order for me to construct M minus? If I have M plus minus, I have the joint machine, then how would I be able to, how much extra information do I have? And then lastly, one thing we can also do to quantify asymmetry is we can consider some notion of distance between these machines. And this is quite a tricky thing to do, even though I won't really talk about how to do it, but we talk about it in our paper, so I won't talk about it so much here. But you can imagine it's a tricky thing to do because M plus has 4 causal states. That means if you write it down as a stochastic matrix, it's going to be, like 8 by 8 matrix, something like that. M minus has 3 causal states, so it's not going to be an 8 by 8 matrix. It's going to be a smaller matrix. So how do you compute relative entropy between 2 matrices that might have different sizes? And so we considered a symmetrized relative entropy for a variety of reasons, but we're able to find a way to do it and make sense of it. So we have these 3 different notions of irreversibility. The first one is saying that are the complexities of the forward process and the backward process the same? The second one saying that how much extra information do I need to know in order for me to infer something about the forward process given the backward process? And then last one is just some metric on the space of hidden Markov models or stochastic matrices. So given these metrics, we applied them to our fly problem, and here's what it looks like. So what we did was We started with a time series. And then, so it's just zeros and ones again. And then we constructed from that a forward epsilon machine. And then we also constructed a backwards epsilon machine. Those are exactly the ones I showed earlier, forwards on top this time. And then we constructed a bidirectional epsilon machine, which is a lot more complex. In other words, there is a lot more structure here than in either one, which is quite interesting. And then we also did that for anesthetized case. And there is clearly difference in complexity there. In fact, in this particular example, the forward and the backwards epsilon machine for the anesthetized case, which is E and F, are identical. But there is still more information in the bidirectional epsilon machine. So we constructed all of this for all the flies, for all the channels. And then we looked at the differences in awake minus anesthetized for each of these cases. So this is all the data we have, and let me explain. Let me take 2 minutes to explain, and I'll wrap up after that. So the left, top left, is the results that I already showed. This is just asking, only considering the forward directional epsilon machine and forward directional statistical complexity, awake minus anesthetized. So that's the data I already presented. And the shaded region in the gray boxes are the regions where there is the difference is statistically significant. In other words, for those channels and for those memory lengths, which is somewhere in, not quite in the middle of the brain, but slightly to one side, we're the signal, there's a significant difference in the signal or statistical complexity. Now, the causal irreversibility, which is top right, there seems to be very little significance. There are 3 regions, but they're not all that significant, and those could be flukes. We don't know. We only have 13 flies. We'd have to do more experiments. So what that tells us is that causal irreversibility between awake and anesthetized, there isn't a huge difference, even though in this picture, it seemed that there was. Now, it was for maybe that particular fly for that particular channel, but statistically, it's not significant. However, crypticity and the KL divergence rates are significant. So in other words, the awake flies tend to have greater crypticity and greater KL divergence rate between the forwards and the backwards direction than the anesthetized one. And it's not, I mean, one might ask, why is that interesting? And I don't have a clean answer for you, but I have an intuition for it. And this is what got us started. And this is very close to what we do in physics. So clearly, you know, there are 2 movies here. One of them is a macroscopic movie of a glass breaking, and one of them is a molecular process. You know that the movie on the left, the macroscopic movie, you know the direction of time. The glass is breaking. That's very clear, the direction of time. But the movie on the right, you have no idea whether it's running forwards or backwards. There's no way to tell. And in physics, we know that the arrow of time emerges at some macroscopic scale, but it's impossible to tell at microscopic scale what the arrow of time is. And that same exact thing should happen in neuroscience. Our brains are somehow already accounting for the arrow of time. And so if I'm watching a movie of a glass breaking backwards, that's going to feel weird to me in my brain, and that's going to feel weird to even a fly, right? Fly is also understanding the arrow of time to some degree. However, if I look at a single neuron, and if I look at the signal in a single neuron, you might not see that arrow of time signal, except if I look at temporal complexity of a single neuron or a single region in the brain, then I might be able to pinpoint the arrow of time. However, that arrow of time information may subside when the subject is anesthetized and not necessarily having a conscious experience. And this is why we think that that temporal asymmetry should be greater for an awake subject versus an anesthetized subject, and indeed that's what we find. So that was the intuition, and I think that's quite an interesting result, because people in neuroscience have been thinking about temporal asymmetry for some time, but they don't quite have the tools to attack that problem. And in physics, I think we know how to deal with that in many ways. So it's in that sense, I think it's been. We hope that it will be a nice contribution to neuroscience. So that's the end of my talk. Those are the results. I mean, it's really applied work, so there's not much more I can say. And here are some funding agencies. And with that, I can say thank you. I think I'll end on this slide, nevertheless. So thank you very much.
Thank you very much, Kevin. That was just a fascinating talk. And sorry, my house doorbell was ringing.
No problem.
Sorry, that was outside my control. Some delivery. No, thank you very much. It was really amazing. And in particular, the spanning from good old Markov and hidden Markov model to the error of time in the brain. That's really a fascinating
Thank you.
journey, and I really enjoyed it. There are not so many of us, so if some of the participants want to ask questions, please raise your hand and I can give you the right to ask your question yourself. Maybe while people think about questions, Kevin, can I ask you one?
Sure.
All these, you know, you mentioned at the very beginning that everything in this talk was classical, yeah, and there was absolutely nothing wrong with that. But some of the concepts that you described could probably easily be moved to the quantum formalism, yeah.
Yes.
Would there be any? Is there any indication that to understand the brain of your flies, that some quantum effect might be at having some role of any kind?
Sure. So yeah, I mean, that's an interesting question. So part of the problem would be that we'd be measurement limited.
Yeah, no, I understand. Yeah.
Now, purely on the stochastic process side, it is possible to generalize the notion of Markov order and hidden Markov models in quantum mechanics, but it's a little bit messy and complex.
Yeah.
Because it becomes instrument dependent and so on. We're actually trying to generalize these notions of complexity to quantum mechanics, but it's been very difficult. There is always some philosophical or technical issue that comes up. So now I think moving it to brain is really, I mean, you know, one would have to think, what kind of experiment can you do so that you would have some data that would have some quantum resolution? And I don't know any. So I mean, something like FMO, for instance, you know, where you can have a claim, even there is not so clear. I don't know how you do that. Maybe one way to think about that is you could ask the question, firing of a single neuron. And I have colleagues here who are using or trying to study the firing of a single neuron using a Bose-Einstein condensate. And potentially there, I should be careful, I don't actually know the details of that, but potentially there, you might get some resolution that's good enough, but even that would be a stretch.
Okay, no, thank you very much. Sorry, I was also just speculating widely.
Sure.
Yeah?
It's
There
kind
is
of
a
fun.
question of Camille that I now ask allowed to talk. Camille, please, you can ask your question.
Yes, thank you very much for this very interesting talk. So just firstly, can you go back to the previous slide?
This one?
Yes, this one, yeah, because you went quite quickly on it. Okay, I just take a picture of it.
Yeah. I've also shared the slides with Francesca and Bethany, so if you like, just get them from them.
Yeah, thank you.
Okay, and yeah, no, I have like a kind of comment question. I saw several times on archive and so on several paper trying to, or at least aiming to describe, I think, consciousness using the theory of quantum contextuality.
Ah.
And also, I think it's also related maybe to what they call theory decision. I don't know if you heard about that and if it might be applied to a fly to try to describe the state of, I don't know, consciousness using contextuality. I don't know.
I think it's, again, you're limited by what kind of experiments you can do. So, I mean, in your contextuality experiments, you have some choices of basis or something like that. And with the flies, you don't have that. You just have these very coarse electromagnetic or electric potential measurements, right? I mean, you can do quite a few different things with the
yeah, no, but I mean, yeah. So I don't know much about it. I hope maybe you heard about it, but from what I know is they. So they apply the contextuality and theory, which I mean first of all came from the quantum information, but they apply that to describe the consciousness, but not at a quantum level, just to, so they ask question to
right.
They make experiment asking question to people, and they see that the brain follow I mean the behavior can be described with this contextuality.
Yes, I think I know what you're talking about. There are people, there's somebody in Australia who does that, and it's not quite quantum, but they take the ideas from contextuality, quantum contextuality, and then translate them into neuroscience or psychology type of questions. And
by itself, it's, I mean, doesn't mean that
of course,
the consciousness is quantum, just a way of describing.
Of course, it just as a motivation and tool. And yeah, I don't know. I haven't really thought about it in that sense so much.
Maybe it could apply to fly. I don't know.
Well, probably not, right? Because maybe because you see that our lives are pretty contextual, even though classical physics is not contextual, right? Because we're, when we're horribly complex things, there's always some context in, you ask me what's my favorite color, and the answer may depend on how I feel. So I think that the notion, there's some notion of context that might arise from complexity, but I think in the truest sense, there's no contextuality there as there would be in quantum theory. But that's an interesting idea, I don't know. I don't have a good answer for you.
Okay.
Thank you, Kevin.
Okay, thank you.
Can I ask sorry. Can I ask a question quick?
Please go ahead, Bethany.
I'm not sure it's very well formulated, and it's possibly dangerous to try to draw empirical conclusions based on physical models, especially as I don't really understand exactly how you have defined time reversibility. But do you think the reversibility of anesthetized brains is a specific effect of the fact that anesthetics seem to target responsiveness in particular, rather than kind of automatic, in some sense, memoryless actions such as breathing? If you look at like how people define lack of consciousness, it always seems to be measured as a response to something. Even anesthetized plants, like if you touch them, they don't close. And responsiveness does kind of suggest some sort of temporal order, action and response. And do you think it would be useful to look at the effects of other drugs that target consciousness to see if this reversibility persists? So yeah, I know they've done they've looked at antidepressants on fruit flies, especially if you want to look at conscious states, different conscious states at the same level of wakefulness. Would it make sense to?
Yeah, I think your explanation is better than what I said. So that's really nice. That's a great hypothesis, and I mean, it would be great to be able to test that. I mean, we should maybe discuss it. I mean, because these guys in Queensland could potentially even do these experiments.
Yeah, I know they've tried the flies on lithium to see, and there's certain. I think they subject them to stress responses, and they get depressed in similar way that depression manifests in humans.
Yeah.
And then treat them with lithium and I think sertraline as well, SSR. So
yeah,
but to see if the time reversibility persists would be interesting.
That's a really great question. I mean, yeah, I don't have a good answer. I mean, it would be nice to be able to discuss that with the neuroscientists as well, where they have some better intuition for the phenomenology of that. But yeah, I mean, you know, one could test, make these hypotheses and test time irreversibility, and see if there is any statistical significance there, right? It would be relatively easy now that we have the toolkit. It's just a matter of feeding the data into the machine. So yeah, it's a great idea.
Yeah, then thank you very much, Bethany, for your contribution, and thank you very much, Kevin, for your suggestion to continue this discussion. And maybe we can do it in another setting in the near future, because that would be really exciting. Yeah.
Yeah.
I'm now looking at the time, and for us here in South Africa, it's lunchtime. For you, it's probably, I don't know, bedtime, probably almost. Yeah. So I think this is now the right moment to thank Kevin for his time and for his really very nice talk. And you know, and nice talks always open new questions. Yeah. And it would be really very nice to keep the connection going, and maybe in the near future have a special meeting to discuss potential of a collaboration and testing some other crazy hypotheses. And I'm sure Bethany will be in touch to arrange it. Yeah,
that sounds great. Yeah, thank you so much for having me. It's been great.
Thank you for taking your time. Yeah, thank you very much. Yeah, so have a good evening, and to the rest of us, have a good afternoon. Yeah.
Have a great lunch. Thanks all.
Thank you, Kevin.
Bye.
Thank you very much. Take care.
Thank you. Yeah. Bye-bye.
Bye.
Bye.